So thank you for coming to Ben's, the thesis defense of Ben Rein. I'll have a few words about this glamorous student. Ben graduated summa cum laude from West Virginia University with a bachelor's degree in psychology and biology in May 2016. Then he was accepted to the UB PPBS program with presidential fellowship. He joined my laboratory in May 2017 and has had a very rapid growth during the past 3 years or so, which is reflected by his impressive accomplishments in autism research. He has published several first author or co-first author papers on high-profile journals, including Molecular Psychiatry, Genes, Brain and Behavior, Nature Protocols, Journal Neuroscience, Translational Psychiatry. Even when the lab was shut down because of COVID-19, he didn't stop working and wrote a review that is published on Trends in Neuroscience. In 2020 alone, he has won 3 awards. He is the winner of Excellence in Research, Scholarship and Creativity Award. He's also the first place winner of 3-minute thesis competition. And he's also the first place winner of Sigma Psi Companions in Zeller's Research Award. Because of his outstanding performance, even during this challenging time of job searching, Ben has got almost every offer he could from the most prestigious schools, including Stanford University, Yale University, Johns Hopkins School of Medicine, Duke University, etc. He has decided to join Dr. Robert Malanka's lab in Stanford University for his postdoc training next March. Ben is a highly motivated, talented, productive, and devoted young scientist, a model student. With his trajectory, I'm sure he will achieve even more. Now let's listen to his full-length thesis defense.
Thank you, Dr. Yan, for the fantastic introduction. I really appreciate all of that. I'm really excited to have my defense here. I'm very grateful to have everyone here. I'm just really excited to share what I've been up to, and been anticipating this presentation for a while, so it's good to finally have it happening. So my thesis, you may have noticed in the flyer, if you saw it, was broken down into 2 parts. The first, let's see if I can get my slides to work, is Synaptic and Molecular Mechanisms in 16p 11.2 copy number variations. And this is a very difficult thing to be working on because it takes so long to say out loud. But I'm sure you're wondering, what are 16p 11.2 copy number variations? Well, on chromosome 16, there's a section which is shown in this little box here. This is just an animation. It's a section of genetic material that contains about 27 genes, and this is position 11.2 on chromosome 16, which is why it's called 16p 11.2. From now on, I'm just going to call it 16p to save myself probably 30 minutes of this presentation. And this entire region of this chromosome is susceptible to something called copy number variations, which is actually what it sounds like, a variation in the number of copies that you have of this region. So the entire section, this highlighted area, can either be deleted or it can be duplicated. It can also be triplicated. There's all different, but these are the most common, the deletion and the duplication. And interestingly, even though you have the kind of bidirectional change here, both the deletion and the duplication are associated with a whole range of neurodevelopmental disorders that actually overlap quite a bit. But before I get to that, I want to use this reference to Down syndrome. This is a very, a lot of people have trouble, understanding kind of this deletion duplication. How can this even happen? Well, Down syndrome, which I'm sure everyone here is familiar with, is caused by a triplication of chromosome 21. So it's actually called trisomy 21. So instead of having 2 copies of this chromosome, you have a 3rd copy. So this is very similar to the 16p duplication in a sense, in that instead of having an entire duplication of a full chromosome, it's just a segment of it. So going back to the deletion and duplication of this region, I mentioned that they're both associated with a range of neurodevelopmental disorders that I'm going to cover in just a moment. So before I get there, I just want to show you, this is the the list of genes that are that comprise the 16p region. If you see your favorite gene in here, congratulations. It has something to do with my research. But the reason I'm showing this is because it's not surprising. It's not terribly surprising that we have so many different neurodevelopmental changes in those with the deletion or duplication, because you have it's not just a single gene mutation here. You're having the change in expression of all these genes. So it can have such a diverse outcome. So in the deletion, these are all the neurodevelopmental disorders and changes that are associated with the deletion. A little key down here, but so in parentheses, what we have is the percentage of those with this deletion that express this phenotype. And you can see that autism occurs in about 1 in every 5. Intellectual disability is pretty common. Epilepsy and seizures is very common. Dysmorphic features and congenital anomalies. Macrocephaly, speech and language delay, the list goes on. We also have another long list of developmental deficits that are associated with the duplication. And you can see that a lot of these overlap. So each of these blue dots represents a common phenotype between the deletion and the duplication. So again, it's kind of amazing to me that whether you have the loss or the overexpression of all these genes, you have very similar changes resulting in autism, intellectual disability, epilepsy, and so on. But there are some unique changes. You can see in the yellow, I think it's yellow, I'm colorblind. These dots here and the duplication, this schizophrenia and bipolar disorder are associated with the duplication, but not the deletion. So there are some unique changes. And then interestingly, there are a few opposing changes where the deletion is associated with macrocephaly, which is increased head size, along with obesity. And the duplication is associated with microcephaly, or reduced head size, and low BMI. So despite these two bidirectional genetic changes leading to so many common psychiatric and developmental changes, they cause opposing kind of physical changes, and that the deletion is increased head size and increased body size in a sense, obesity, and the duplication is reduced head and body size. So let's talk about the prevalence and the treatment of these copy number variations. So this table, as Dr. Yan mentioned, during the coronavirus quarantine, I spent some time writing a review article, and I did a meta-analysis of all these clinical reports studying these copy number variations. And so I looked at in general population and in clinical cohorts, or within populations that have some sort of neurodevelopmental disorder, whether it's autism, intellectual disability, and so on. And I found that on average, or approximating, 16p 11.2 deletions occur in 1 in every 2,300 to 3,500 children in the general population. So you've probably never heard of this before, I would imagine. But if you've heard of narcolepsy, that's a sleep disorder that I actually have. It's about 1 in every 2,000 children. So I have that. I was almost equally likely to be born with this sleep disorder as I was with a 16p deletion. So it's not terribly uncommon. But if you look at those children with some sort of neurodevelopmental disorder, like I mentioned, 1 in every 220 or so of them have the 16p deletion. So it seems to be pretty prevalent within affected populations. Now, if we look at the duplication, the numbers are pretty similar, give or take. And again, these are estimations based on kind of a summary of published data. But when we talk about treatment strategies, if you want to take a guess, how might these patients with these duplication or deletion be treated? You guessed it, there's no treatment strategy available because we know so little about how these genetic changes drive the developmental changes. And it makes sense because there's so many changes or so many genes that are being altered. It's such a complicated system to be studying. So here's where my research kind of comes in. In 2011, Dr. Aaliyah Mills at Cold Spring Harbor generated mouse models. This is just a figure showing some chromosomal engineering. She generated mouse models that carry the 16p 11.2 deletion or duplication. So she created 2 mouse models. I'm just showing here the labeling. So the deletion mice have 1 normal copy and 1 deleted copy. So the plus minus, this is going to be labeling for the deletion throughout the paper or throughout the presentation, sorry. And the duplication, they have 1 duplicated copy and 1 normal copy. So having these mouse models enables us to ask a very important question, which is how do 16p 11.2 copy number variations affect the brain? By the way, I meant to say this in the beginning. I'm going to say it now. I designed this presentation in a way that hopefully those who aren't, don't study neuroscience or aren't in my particular field will, I try to keep out too much jargon and so that, you can follow along. So I want everybody to kind of do their best to stick with it. I know it's easy to get lost in a Zoom meeting. So, and I guess if you have questions, you can interrupt me. So anyhow, these mice allow us to ask how these copy number variations affect the brain. And when I first joined the lab, I was really excited, very eager to get involved in some research. And Dr. Yan said, hey, Ben, we have these 16p duplication mice, and there's really nothing known about this system at all, or this model, or how it happens. We don't know anything about it really. So she said, do you want to study this? And I said, absolutely. This sounds really cool. I'd love to. But the first question I had to ask before doing anything else was, do these mice display behavioral changes that are similar to those that we see in humans with the 16p duplication? Because if they don't, there may not be any value in studying how their brain is changed. So, the social, cognitive changes, things like that. So this all began with a behavioral characterization of the mice carrying the 16p duplication. This is sort of a label up in the top here. And then for the bars, there's going to be some bar graphs, and this is the color key. So the first test I did was the 3-chamber social preference test. And I'm going to talk a lot more about this later on, but basically what this test does is you put the test mouse in the middle of this apparatus where on one side there's a mouse under a cup and the other side there's an object under a cup. And the mouse has the opportunity to freely explore the apparatus and engage with the mouse or the cup or the object, whatever they want. And what we typically see is that the wild type mice, so you're going to see this a lot too, WT, this is wild type. This is just control mice that do not have the genetic mutation, the 16p duplication. So these are always going to be our control. You're going to see this on every figure. If we look at the amount of time they spend interacting with the social and the non-social stimulus, the wild type mice show a very strong preference for interacting with the social cue. So this is measuring social preference. The mice should spend more time interacting with the other mouse. But when we look at the same behavior in the 16p duplication mouse, we find that they don't show the same social preference. So there's actually a loss of social preference. And in addition to that, comparing the amount of time that the wild type mouse spend interacting with the social cue versus the duplication mouse interacting with the social cue, there's a significant difference. Also, again, for my friends and family watching, if you see 2 bars highlighted with this, kind of like bracket and then some asterisks, that just indicates that there is a statistically significant difference between these values represented by the bars. So here you can see that there's the duplication mice have significantly lower social interaction time indicated by this bar here, this bracket. So they do have some social preference deficits. Now, what about, oh, I'm sorry, I forgot to say this. This is a social preference index, which basically just condenses all this information here into one value, and it is significantly reduced. So they do have social preference deficits. But what about temporal order, or what about cognition? In order to measure or to assess the cognition of these mice, we used a test called temporal order recognition memory. And in this task, the mouse is first exposed to 2 identical objects. Then an hour later, they're exposed to a different pair of identical objects. Then 3 hours later, they're given one of each. And you can see that the mouse is spending more of its time in this graphic with the first object it saw. And this is because mice prefer novel objects. They enjoy novelty. And in this case, they were exposed to this object 4 hours ago and this object 3 hours ago. So it's basically assessing their ability to access recently obtained memories and kind of juggle them. It's called working memory. So when we test the mice in this paradigm, you can see this is just a discrimination ratio. It's just a value that assesses how well they performed. You can see that the duplication mice performed significantly worse than the wild type mice. So it looks like these mice do in fact show social and cognitive deficits. And what's very interesting about this is that both of these tasks both involve the prefrontal cortex, which is a region of the brain. So in humans, the prefrontal cortex is a very advanced brain area. It's involved in high-level executive functions, decision-making, things that are difficult to do and take a lot of brain power, you would think as. It's also involved in social behavior, personality expression. And if we were to zoom in and look at the prefrontal cortex, there are 2 main cell types. And as you may know, the cells in the brain interact with one another and junction at synapses where they communicate and pass neurotransmitters, which activate receptors. So there's 2 types of cells. Like I said, the first is pyramidal neurons. And these are labeled in green because they release a neurotransmitter called glutamate, which is excitatory, meaning when the first cell at the synapse, the presynaptic cell, releases glutamate, it activates postsynaptic receptors, in this case, NMDA and AMPA receptors, and this causes the second cell to activate. So there is activating and inhibiting neurotransmission in the brain. On the other hand, we also have interneurons, and these release a substance called GABA, which inhibits the next cell in line. So this is a little bit, complicated. So I want to kind of zoom in and look at one single cell, and I can kind of explain to you how these two forms of neurotransmission balance together. So this circle is meant to represent a cell, and you could see it's expressing some NMDA receptors. So let's just pretend in this model here, this is a sort of a simplified model of a cell. And I'm just saying that because it is simplified. If there's glutamate expressed or present outside of the cell, this is going to cause these NMDA receptors to activate, which will cause calcium to flow into the cell. So these are actually ion channels. When they're activated, ions flow into the cell, and this causes the cell to become more or less excited. In this case, calcium is a positively charged ion, so this will cause the excitation to move up. If the cell is also expressing AMPA receptors, the glutamate will similarly activate these, and this will cause sodium to flow into the cell, further driving the cell's activity upward. Now, this is not good. The cell does not want to be too excited or too active. This can cause toxicity and cell death. Instead, it wants to be kind of right here in the middle at this homeostatic point. So luckily, we have the expression of GABA receptors, which, when activated by GABA, cause chloride to flow into the cell, which it's negatively charged, and it will cause the cell to return to its normal excitability. Great, it's happy again. But this balance can be thrown off in either direction, where if you have too much GABA extrasynaptically or in the synapse, and let's say you have the loss of some of these NMDA and AMPA receptors, now the cell's receiving too much inhibition and its excitation is too low. So this excitation-inhibition balance needs to be maintained on an individual cellular level for those cells to be functioning in a healthy way. So the cool thing is, in Dr. Yan's lab, we have the ability to measure the currents that are mediated by basically the activity of these receptors through a method called slice electrophysiology. So what we do, I'm crediting Megan, because my lab mate, because she showed me this cool graphic that's very useful. We can take brain slices and incubate them with artificial cerebrospinal fluid and oxygen to sort of simulate the conditions of the brain and preserve the cells, keep them alive. Then we can put these cells or these brain slices under a microscope and image the cells. And if we look at these cells, we can actually find one, let's say this one. We have a recording electrode that we can sort of implant onto the cell. And from there, we can record these electrical signals that are coming out of the cell. So we could just record this cell, how it's functioning normally, or we can also put a stimulating electrode over here and stimulate some upstream cells and record what happens to this cell in response. So we wanted to look at, since these mice showed these behavioral changes related to the prefrontal cortex, we wanted to test, do these mice show any sort of synaptic deficits in the medial prefrontal cortex? So we first looked at NMDA receptor mediated excitatory postsynaptic currents. So basically, like I said here, we're stimulating some cells and we're recording the postsynaptic currents in this cell that are mediated by NMDA receptors. So you can actually see this is an actual trace of, we're looking at current flowing through the cell, through the ion channel, and we're looking at the amplitude. So, how much current is passing through these receptors. And you can see that the duplication mice compared to the wild type show pretty much no change. And when we actually quantify it, looking at various stimulation intensities, because the stimulation can be weak, can be strong, we kind of do a stepwise increase. We see no change really in the amplitude of these NMDA receptor signals. But if we look at GABA, you can actually visibly see that there's a reduction in the amplitude. And when we plot this out again, you can see there's, again, these asterisks indicate significance. You can see there's a significant reduction in the amplitude of these GABA currents. So again, GABA is inhibitory. So the loss of GABA would maybe cause the cell to be more active, potentially. An electrophysiologist would disagree with me, but an electrophysiologist will see that this is true based on this study. Anyhow, so we next looked at evoked action potentials. So when cells are active, when brain cells are active, they fire action potentials. And we can measure the frequency of these in these cells. And you can kind of tell here that the frequency in the duplication mice appears to be faster. And when we actually quantify it, these cells are firing more action potentials, which indicates that they are more active or more excitable. So since we saw the loss of GABA and we saw the increase in action potential frequency, I thought maybe this is caused by a loss of those interneurons. Remember, I showed you before, these are the cells that provide GABA onto these cells that we're recording from. So I can actually label these cells and image them under a microscope with immunohistochemistry. And what I did was I labeled paravalbumin expressing interneurons. This is just a subtype of interneurons, and actually counted them in two areas of the prefrontal cortex, the cingulate and the prelimbic cortex. But I found that there's no change between the wild type and the duplication mouse. So it's not actually that there's a loss of these interneurons. It must be something else. So at this point, we decided to use a technique called RNA sequencing to get to the bottom of this. As you may know, DNA is transcribed into RNA and then translated into protein. And what we can actually do is we can measure the RNA level of a bunch of genes all at once with RNA sequencing. So we actually take PFC tissue, isolate the RNA from it, run it through a sequencer, and we can look at the expression level of every single gene in that tissue that we provided. And what we can do with this is we can compare the mice with the duplication to those without the mutation and look at what genes are being expressed at different levels. And that maybe could give us some clues into what molecular mechanisms are responsible for the GABA changes that we see. So when we did this, we find that a molecule, so this is actually just a spreadsheet showing the top 18 downregulated genes. So these are genes that are expressed at lower levels in the duplication mouse. And number 18 here, NPAS4, caught my attention because it is a gene that regulates GABAergic synapse formation. And what it actually does, it's an activity-dependent transcription factor. I'm sure I just lost a few people, so let me explain what this actually does. So in a wild-type cell, again, here we have our cell with our nucleus and our excitation continuum. When NMDA and AMPA receptors are activated, calcium and sodium flow in, cell becomes activated. Now, like I said, NPAS4 is activity dependent. So now that the cell's activated, we're going to see NPAS4, it's unhappy. We're going to see NPAS4 expressed in the nucleus. What's going to happen now is NPAS4 is then going to do its function. It builds GABA synapses, and it's going to form more GABA synapses, which will in turn allow chloride to flow into the cell and restore the homeostatic excitability. So what NPAS4 really is doing is it's preventing the cell from staying at an unhealthy hyperactivated state by functioning in this kind of feedback loop. So in the duplication then, if we have the loss of NPAS4, what we would expect to happen is that the cell's activated normally. We know that NMDA receptor activity is intact. I already showed that data. So the cell becomes activated, but instead of NPAS4 being expressed now, it's not for whatever reason. As a result of this, what we would expect is that there's no GABA synapse formation. This would potentially explain the GABA loss. There's no GABA synapses being formed or fewer, then this could explain why there's a reduced amplitude of these GABA currents. And if the cell's staying up here at this hyperexcited level, this could explain why the cell's firing more action potentials. So it seemed like a plausible hypothesis that NPAS4 is kind of at the root of this. So let me take this whole hypothesis and put it over here on the side. So reduction in NPAS4 is potentially leading to fewer GABA synapses, which is promoting the GABA deficits and the hyperactivity and leading to social and cognitive deficits. So the very first thing we had to do to verify this theory, whether it was true or not, is to test whether NPAS4 was actually downregulated or whether this was maybe just some sort of artifact that's inaccurate from the RNA sequencing data. So looking in the mouse prefrontal cortex, used a technique called qPCR to measure the mRNA level of NPAS4. And this is clearly, you can see there's a lot more samples here. There is a significant difference. So we did verify that NPAS4 was being expressed at lower levels at the RNA level, and then also looking at the protein level, it's also reduced. So now notice on the left here, we have this check mark flying in, bingo, NPAS4 is downregulated. Before I move on to this next stuff, I just want to add, I had a unique opportunity at this stage in the project to actually investigate whether NPAS4 was similarly downregulated in the prefrontal cortex tissue from human children with autism. And interestingly, we actually did find that the same reduction was occurring in human patients with autism. And this is not just those with the duplication, this is multiple forms of autism. So this indicates that potentially the loss of NPAS4 may have a broader role in autism in general, not just in this one mutation. Now, anyhow, focusing on this theory I have. So if we're going to really focus on this, I figured it's an easier way to, instead of verifying all these things, sequentially, what if we could actually increase the level of MPAS4 and test whether this is sufficient to build more GABA synapses, which would then restore the GABA function and the normal PFC activity and improve the social and cognitive function. And if this is the case, then could MPAS4 maybe be a therapeutic target? Remember in the beginning, I said there's no treatment strategies available. Maybe we can figure out the mechanism underlying these changes and also build a potential treatment strategy in the process. So again, first thing I wanted to do here, I'm replacing my old theory with now this new theory. First thing I want to do is increase the level of MPAS4. And so the way we did this was we can actually drive MPAS4 overexpression by injecting an overexpression virus into the prefrontal cortex in live mice, and they're perfectly intact afterwards. And then we can actually measure their behavior and do all sorts of experiments afterward. It doesn't affect them at all. But I wanted to make sure that the virus was actually working and increasing the level of MPAS4. So I did some staining here, and this is in the prefrontal cortex. And you can actually see, this is pretty zoomed out. Each of these dots is one cell. And you can kind of see that a lot of these cells are expressing MPAS4 at a much higher level. So these bright dots. And when we quantified it, we did find that it was increased. So yes, okay, step one, MPAS4 is overexpressed. Now the next question is, does this lead to more GABA synapses being formed? So a GABA synapse might look like this, obviously simplified, but we have a GABA receptor on the postsynaptic side, and on the presynaptic side, we have these vesicles containing GABA that are released, and they're actually, the vesicle that carries GABA is called VGAT. It's a specific vesicle for GABA. And what we can do is we can actually image VGAT under a microscope and use that as a marker for the presence of a GABA synapse at that location. So this is a lot to look at. Let's just start by looking right here. So this is in the wild type mouse treated with GFP. So GFP is just a control virus. It doesn't do anything. So this is just a control. The red dots represent VGAT. So you can see there's all these GABA synapses all over the place. And for reference, the blue circles, the larger blue circles, are cell nuclei. So you can see that these red dots are little and they're everywhere. But when we looked in the 16p duplication mice, there's clearly much less expression of VGAT. So there's far fewer GABA synapses. But when we give these mice this MPAS4 injection, it actually, you can see that it causes an increase in the expression of VGAT. And when we quantify it, there is a significant increase. So it does appear that overexpressing MPAS4 does in fact cause more GABA synapses to be formed. Now, the next question is, does this restore GABA function and PFC activity? So this is, again, these figures get really heavy to look at when we have so many groups, but you can pretty much just look at the light blue, and this is actually a dark blue. So this is the activity of GABA before the MPAS4 treatment, and then this is after. And you could see that after the treatment, the 16p duplication mice have significantly increased GABA activity, which makes sense because we have now formed more GABA synapses and they are now functioning in the cell. In turn, we find that, again, looking at the light blue and the dark blue, the action potential firing frequency goes from being way higher than the wild type mouse to being pretty much very similar, a lot closer. So it definitely brings it down. So this would suggest that by restoring the GABA synaptic activity, we are therefore restoring the excitability to sort of a homeostatic level. Now, the big question after we verified that is, does restoring the function of the PFC synaptically improve the social and cognitive performance? So going back to this 3-chamber social preference test, we did find that, again, wild type versus 16p, there's a significant loss of social preference. But after we give these mice this MPAS4 overexpression, it restores their sociability, and you can see they're almost indistinguishable from the wild type mouse. So this made a very substantial impact on their social performance. And actually, this came very far into my project, and I refused to believe it. This was not the first sort of restoration experiment I had tried. And I went back and I purposely gave the injection of the virus in the wrong location to verify, and it didn't help. I tried all different things. And even after the paper was published, I went and did the experiment one more time just to make sure. And it actually does significantly increase their sociability. Now, what about their cognition? Again, the same temporal order recognition memory task. They show significant impairments at baseline, but with this treatment, it enhances their performance. So this is pretty crazy. It looks like restoring MPAS-4 does kind of go and check all these boxes and result in improved sociability and cognition. So this is sort of the conclusion of this project, but I want to answer this other quick question. What about the mice with the 16p deletion? Maybe from the beginning you were wondering, what happened to them? In the beginning of my PhD, I partook in a study that investigated these mice as well. And interestingly, in the prefrontal cortex, again, we measured action potential frequency, and you can see that there's a significant reduction. So the prefrontal cortex neurons of the mice with the deletion actually have a reduced activity, which again is the opposite of what we saw in those with the duplication. We wanted to test this is kind of complicated, but this is the prefrontal cortex. We infected these cells with an artificial receptor called a DREAD. And basically what this allows us to do is we can put this receptor onto the cells, then we can activate the receptor and cause the cells to become more active. And when we did this, it did in fact increase their activity. And the question we wanted to know was, is this loss of activity in these cells responsible for the social and cognitive deficits that these mice show? So when we test the social approach task, which is, as you may assume, a social task, these mice, you can see they show loss of social interest. They have significantly lower levels of social interaction time at baseline. But when we activate these receptors and cause these cells to become more active, they spend significantly more time interacting with the social stimulus. Similarly, in a task called the Barnes maze, which is a memory task, it makes these mice perform significantly better. So in summary of part one, we know that 16p copy number variations are associated with a bunch of neurodevelopmental disorders, but the mechanisms are very unclear. But we found that in the duplication mice, they have these GABA deficits and increased neuronal activity. Whereas in the duplication mice, they have reduced neuronal activity in the prefrontal cortex. And in either model, restoring homeostatic neuronal activity reverses the social and cognitive deficits. So this suggests a model where we have PFC activity on a continuum, and on the high end, we have the 16p duplication. The cells are too active, and this is, like I said, this is bad. This causes the brain area to not function properly. On the other end, we have the deletion that are firing kind of hypotonically on a lower level. They're not functioning, they're not active at the level that they should be. But in either case, the deletion or the duplication, restoring the excitability to the median homeostatic level is sufficient to improve their social and cognitive deficits. So like Dr. Yan mentioned, I wrote that review article during quarantine, and from reviewing all the literature on this field of neuroscience, there is no part of the brain or any sort of molecular mechanism that shows this clear of a bidirectional change. So this hyperactivity, hypoactivity kind of theory in the prefrontal cortex it's kind of an interesting theory for why the duplication and the deletion, both, despite being such different changes, such a huge amount of genes, how they both lead to social and cognitive dysfunction. So yes, there is a part two. I'm not done yet. This part's much shorter, don't worry. But this part is called Advancement of Behavioral Assays for Sociability in Mice, which is what it sounds like. So as you can tell by the first part of the presentation, my research is very focused on autism spectrum disorder. And as you may know, autism is characterized by deficits in social communication, as well as restricted or repetitive patterns of behavior. So there actually are a lot of mouse models of autism spectrum disorder, which may be surprising if you're not familiar with the field. There are transgenic mouse models. So these are mice with, I'm not going to go through this whole list. I'm just showing you some examples, but mice with single gene mutations or copy number variations like the ones I study. So there's all sorts of different genetic mouse models. There are environmental mouse models, drug-induced mouse models. So many models of autism, but there's kind of this overarching question of how do we evaluate social behavior in mice? That's when I talk to my friends and family about my research, that usually that's their first question is, how do you even look at that? So the 3-chamber social preference test, this is what I mentioned in the very beginning. This is the premier method of measuring mouse sociability, developed by Dr. Jackie Crawley from UC Davis. She visited last year at UB. We got to talk to her, which is cool. And in the process of preparing these papers and doing all this research and reviewing the literature, I found that there was a bunch of different protocols of this test across different papers. I found 6 or more, at least 6 different protocols, which you probably are thinking, okay, so what? Well, the different protocols that were used resulted in different conclusions. Actually, these are 3 different mouse models of autism. For all 3 of these, there's one scientific paper showing that they do have social deficits and one showing that they don't have social deficits. And this is important because, first off, we need to know whether these mouse models are valid, but also these papers that are presenting inconsistent results are in top journals like Nature, Science. And if the protocol that they're using is resulting in totally different, conclusions, that's a problem. So we decided to propose a standardized 3-chamber social preference test. And in order to do this, we first had to look at the original. So this is the protocol proposed by Dr. Jackie Crawley. In this protocol, the mouse is first habituated to an empty apparatus for 10 minutes. Then they're put into this apparatus. Like I said, earlier I mentioned this, there's a social stimulus, which is a mouse under a cup, and they've never seen this cup before. On the other hand, they have a non-social stimulus, which is just the cup. So looking at this, there's one thing that jumps out at me, which is if they have a choice between interacting with this or this, you have either a mouse and a cup or just a cup. It seems almost obvious that they're going to prefer this stimulus because they've never seen either these mouse or the cup. And it's basically 2 versus 1. They're going to spend more time over here. So there's kind of this inherent bias favoring the social stimulus, and that may potentially mask the presence of social deficits because it reduces the overall sensitivity of the test. The other thing is that there's no habituation to the cups, which makes sense because they're using a cup as the non-social stimulus, but the mouse may enter the chamber and see this right here and run over and start interacting, and it looks like they're having a social encounter, but they're actually sniffing the cup because they're interested in the cup. So you don't even know. And this could lead to unplanned, unpreventable variability in the results. So what we did is we proposed a modified protocol of this task where the mouse is first habituated to the empty cups to just kind of familiarize with them and sniff them and get used to them. Then we put an identical object under each cup. In this case, we use a crumpled up paper towel ball. It's just a generic, easily available object that they can smell and interact with. And the purpose of this is for them to become familiar with the presence of an object within the cup that is distinct from the cup. Finally, they're able to interact with the mouse under the cup or a novel object under the other cup. So we have a true non-social stimulus to compare with the social stimulus. And in this case, the cups have nothing to do with it. The mouse is just interacting with the mouse or the object. So we wanted to test whether or not this was effective at, testing social deficits. So we looked at an array of mouse models of autism. And so I'm just going to put up some data here. Shank3 mice are Shank3, 16p, and Call3. These are all genetic mouse models of autism. Don't worry about the details. But when we tested the mouse, the Shank3 mice with the original protocol, this one here, we find that they actually don't differ significantly from wild type mice. But when we test them in our proposed protocol, they do show significant deficits in social preference. So it does appear that our protocol is a little bit more sensitive to social deficits, which makes sense because we're getting rid of that inherent bias that favors the social stimulus. Again, we found the same exact results in the duplication mice, same results in Call3 mice. And then lastly, we tested D4 knockout mice. These are mice lacking the D4 dopamine receptor. These have very little, these are not an autism mouse model, but we wanted to test them as a control for looking at a mouse with a genetic modification without autism. We want to make sure this test isn't causing false positives. And we do see that there's no significant difference. So our task is sensitive at detecting social deficits and also reliable. It doesn't fall for false positives. So after kind of getting to this point, this is published, but I had a question that I was thinking about, which was, how can we further stratify mouse behavior, mouse social behavior? I'm really interested in social behavior. I'm really interested in kind of, Dr. Yan mentioned my undergrad was in psychology. And I tend to think about, these behavioral tests that we use. So we can measure social preference with a 3-chamber task. We can measure social interest with just a general social interaction task where they just interact. But what about social motivation? We really can't measure this. And of course, you would imagine that social motivation must be some sort of meaningful component of autism spectrum disorder. So I was thinking about how we can measure this, and I came to this idea of using the elevated plus maze. The elevated plus maze, as you can see, is an elevated plus shaped maze. 2 of the arms are covered by walls and 2 of the arms are open. And this is used to evaluate anxiety or anxiety-like behavior in rodents. So what you do is you put the mouse in the chamber or in the middle here, and then after you let them explore for 5 minutes, the mouse is going to naturally prefer the closed arms because they're protected, they're dark. This is what mice like. They like to be in tight little spaces that are dark. On the other hand, the open arms they're very aversive. They're bright, they're open, they're elevated. So this is, relative to this arm, this is much scarier for this mouse. So what you do when you measure anxiety is you put them in for 5 minutes and you measure the amount of time they spend in the open arm. And the less time they spend exploring the open arm, the more anxious they are presumed to be, which makes sense because if they're more anxious, they don't want to go out here in this scary environment. But I thought about this, how can we use this to measure social motivation? So I thought, what if we took our wild type and our autism mouse models, and we first tested them on a baseline trial, just a normal, just like I just showed you, elevated plus maze, to make sure that there's no differences in anxiety. Once we verify that there's no difference there, we can then test an object motivation trial, where we put a object under a cup in the open arm, and we test, is this sufficient to coax the mouse out into the open arm? Are the mouse models of autism willing to come out here into this uncomfortable environment to interact with an object? And then similarly, are they willing to come out here to interact with the mouse? So this kind of gives us an ability to look at the animal's motivation to interact with an object and a social cue. So what we find is that in the baseline trial, here we have the closed arms and the empty arm, or the open arms. When the open arms are empty, the wild type and these 2 mouse models of autism, SHANK3 and BTPR, they all spend their time in the closed arms because they have no reason to go out into the open arms. It's scary out there. They don't want that. So they stay in the middle. They just, hang out. But when we put an object into one arm and keep the other arm empty, they spend very little time interacting with this arm or going out there and exploring. But interestingly, they all spend a lot more time out here in this arm exploring the object. Now, when we put a mouse in the open arm, unsurprisingly, we find that the control mice, the wild types, spend more time out here exploring the social arm. But the SHANK3 and the BTPR mice, the autism mouse models, they don't go out into this open arm. They spend significantly less time out here, which suggests that while their motivation to come out here and interact with an object is intact, their motivation to come out and engage in a social interaction is somehow deficient. So in summary of part 2, the existing methods for evaluating sociability in mice are insufficient. There are some imperfections with the way some of them are designed. The proposed 3-chamber social preference test protocol displays greater sensitivity, and hopefully by standardizing it and providing this protocol for the field, it'll enable more future studies of autism mouse models to be more consistent and get rid of those issues that we saw in the literature. And finally, the modified elevated plus maze that I created here enables the evaluation of social motivations. So hopefully future studies looking at social motivation will maybe use this. And maybe more importantly, we find that in autism mouse models, they have this deficient motivation to interact with the mouse, but not an object. So interesting stuff. I came up with this idea. I thought, I'll test it, and it ended up working out, and it was just good luck, but it's cool. And I'm really interested in the development of behavioral assays. And I hope that maybe these 2 projects will give me a little bit of clout in the behavior field so I can continue to make more assays like this in the future. So with that, on to my acknowledgments. I want to first thank Dr. Yan. Dr. Yan, you've given me tremendous help and training and advice throughout my PhD. I'm very, very grateful for all the opportunities I've had to just, your door's always open. Every time I feel like I'm lost, I talk with you, I come out of there feeling like I'm completely oriented. So thank you very much for everything. My committee, Dr. Sim, Dr. Slaughter, and Dr. Paez, thank you so much. I know that serving on the committee is optional, so I appreciate you taking the time to further my development as a scientist and pushing me to think about my research in new kind of ways that I typically wouldn't be able to. I have a long list of people to thank in the lab. Ms. Chen, if you're on this, thank you so much for everything you do for the lab. You are the foundation of our lab, and I wouldn't have been able to do any of the studies without you taking care of our mouse colonies. So thank you so much. Many people to thank from basically the rest of this lab. Some of these people are still in the lab, some of these people are gone, but so you know who you are. You've done all of my training, all of my ideas, my troubleshooting, creative discussions, everything. Thank you. I love all the members of our lab. It's been awesome being a part of this lab, and I really think we have such great people. So thank you for making me feel welcome and always being there to help me out. Let's see here, my computer's failing. To support, or in support of me, were my family and friends. So thank you to my girlfriend Bella down here, who listened to my presentations a million times. She was always my first audience. My family, obviously, for being tremendously supportive. My friends for always being there when I needed a break outside the lab. The 2016 PPBS class, thanks for all the good times in the beginning. And, we got through a lot of classes together. And then lastly, but not least, thanks to Michael Greenberg for the MPAS4 antibody, Leah Mills for the duplication mice, the genomics and bioinformatics core for the RNA sequencing, Wade at the confocal microscopy core for all the help with the imaging, and then the Nancy Lurie Marks Family Foundation and the NIMH for funding. And with that, I will open the floor for questions. Thank you.
Great job, Ben. If you don't mind, I have a couple questions.
Steve, go ahead.
So one of my questions was, can there be partial duplications or partial deletions of the 16p 11.2 region?
Yeah, great question. Yes, there can be, there's about 27 genes in there, and you can have, duplication or deletion of like 23, 24 genes. There's actually, if you look at the studies and compare them, I say about 27 with a little wiggly mark, because some of the papers say 26, some say 29. So there's definitely, sort of differential expression of this entire region. So yeah.
Okay.
And that is maybe a big question of like, how does that change the potential outcome? So yeah.
All right. So I don't remember the task, the task that you used, what it was called, but the task requiring new stimuli. Why did you use that particular task in the place of you said it was the Barnes maze? You switched tasks for cognitive evaluation between your duplication and your deletion.
Great question, Steve. So the deletion, I presented it after. Yeah, the deletion I presented second, but the deletion paper actually happened first. The Barnes maze is more of a hippocampal task that involves prefrontal cortex. So that was kind of already done when I joined the lab. But the reason I decided to use temporal order instead is because there's a paper showing that if you lesion the prefrontal cortex in mice, they are unable to perform that task. So it's been basically isolated that the temporal order task that I used is specific to prefrontal cortex function. And that is the region of the brain that we typically study. So it seemed like a good place to start. And we ended up finding that they did show deficits. So there's really no good explanation to answer your question of why we switched, but at least I have the explanation for why I chose temporal order.
It makes sense. Thank you. And the last one, so you tested for comorbidities with anxiety to get a baseline of that. Why didn't you? And I noticed in both deletion and duplication, you have depression as a possible effect of it. Did you ever think of testing for depression with those mice?
Yeah, so I show in the presentation social and cognitive behavior, but I actually tested a whole bunch of things that I didn't show. Including anxiety, schizophrenia, motor delays, Actually, here I have supplemental data here. Let's see. Yeah, novel object, another task. Yeah, elevated plus maze. So anxiety. So yeah, the reason why, I'm not going to go all the way back. The reason why I wanted to do the baseline trial was, I guess it doesn't really matter if they have some sort of other depression phenotype, as long as they don't differ from wild type in their baseline performance on the elevated plus maze task, because that's evaluating one specific facet of anxiety or depression. And as long as that their function in that, or their behavior in that test is not affected, then it shouldn't affect their performance once an object or a mouse is introduced.
Okay. Thank you.
Thank you, Steve. Good questions.
I have a couple questions, if I can go.
Hello, Will.
Hi. And so first of all, great presentation. That was awesome. Very easy to follow. So my questions are on part one. So I'm just wondering, and I apologize if I missed this, I had to step away for a minute, but do you know of any therapies that exist now that might be able to alleviate that NPAS issue that you identified?
So what you're asking is what I'm working on now, because yeah, it seems like MPAS4 is a really effective target molecule. So, yeah, like, is there anything that's FDA approved that we can just increase MPAS4? The answer is no, and it's complicated. So MPAS4, like I mentioned, is activity dependent. So the only way to increase MPAS4 expression would be to increase activity with something like, let's say, Ritalin, methylphenidate. But the problem is that the cells are already hyperactive. So if we further that, it's going to be problematic, especially because the duplication is also associated with epilepsy. So it could potentially exacerbate that problem. But I've been looking at molecules that have been shown to maybe function upstream of MPAS4, and I did find HDAC5, histone deacetylase. It's an epigenetic enzyme, which also our lab studies epigenetics, so it works out great. It regulates MPAS4, so I'm trying to inhibit HDAC5 using a small compound called LMK235.
Oh.
So, yeah.
And then one other question. So you settled on NPAS4 from that. There's a whole list of them. Do you have any other kind of theories that of any of those other genes that might be involved, or have you not really investigated that?
So I made it look really simple. Like I just found MPAS4 and it was all, golden, but I actually, I investigated a lot of different proteins and molecules before I got to MPAS4. I did qPCR for like 30 genes and spent all the lab's money on Cyber Green, unfortunately. But yeah, there's other molecules that seemed plausible, and I tested some other stuff and it didn't seem to work out. Do I think there are other molecules involved? Definitely. NPAS4 is not one of the 16p genes, so there's no clear direct mechanism that would drive its downregulation. And even HDAC5, like I mentioned, also doesn't have anything to do with the region. So there's got to be some other things at play. Being able to identify exactly what that is, stemming from the downstream downregulation or down or upregulation of 27 genes all simultaneously, is probably going to be impossible, but or at least very painstaking. But yeah, I'm sure there are other molecules involved, but I'm satisfied with settling at MPAS4.
Yeah, good job, man.
Thank you.
Ben, can I ask you?
Yeah, of course.
Yes, well, very good, very good. I'm really impressed. I knew it would be so good, but I didn't know it would be just really amazing. So
thank you.
I'm actually trying to see more like not specific about your experiments, but your sort of vision of this CNV models in general. As probably your supervisor, Dr. Yan, she knows well that the story with a lot of autism models started with single gene models. Now, seems like CNV models might provide even, very different, not very different, but much more complicated picture of this disease or pathogenesis, or at least some aspects. And one of the more probably pervasive difficulty with this model, or difficulties of with these models, is then, this CNV is present in every single cell of the body. And I know you understand this, appreciate this, but what do you think? There's all your abnormalities. how, you registering or recording from the cells that you sort of, have some ideas that could be explained by excitatory or decreased inhibitory, but in fact, all cells are abnormal, right? So
Yep.
what do you think, how we could sort it out, or at least starting sorting it out?
That's a good question.
You go into Robert Malenko. If you had an idea, you would be the star there too.
Yeah, well, so, okay, let me just make sure I understand the question. Are you saying how would we isolate the, body-wide, organism-wide, or?
Well, no. So there's both interneurons and pyramidal neurons, and all other types of neurons in the brain have this deletion, or a chromosomal abnormality, a duplication. So how would you think we could figure out what comes first, what comes second, or they all work together and it's a mess and we can't really sort it out? Do you have any idea? Did you discuss it with with Dr. Yan about possible sort of, because I think it's not, not just for this model. There's a lot of other, like 22q deletion, which is very popular model for schizophrenia. And that's sort of what we see everywhere. We don't have a good approach yet. I mean, I'm sure there are smart people that are thinking about this, but do you have what you think? What's your personal take on that?
Yeah, I mean, from reviewing the literature, I already know that there are differential electrophysiological and molecular changes throughout the brain. There's a bunch of brain areas, not just the prefrontal cortex. So, and they're all affected differently. So I know that it's not just the PFC, but I guess, you gave me an idea or perhaps a hint that, it would be cool to maybe do a cell type specific knockout or duplication of, the 16p region in interneurons and in pyramidal neurons, because MPAS4, which you actually may be familiar with, I'm assuming, because your student's presentation, they were talking about MPAS2 or MPAS3. MPAS4,
yeah.
MPAS3, yeah. MPAS4 functions not identically, but in the same overall way, I guess, in both interneurons and pyramidal neurons. So in pyramidal neurons, when it's overexpressed, it causes more GABA synapses. But in interneurons, when it's overexpressed, it causes more glutamatergic synapses onto the interneuron to then promote more inhibitory input onto the pyramidal neurons. So I suppose I've always used that as my out when people ask this type of question. It's like, well, it's probably changed in both cells because either way it'll cause the same change. But it would be interesting to see, like if we delete it or duplicate it only in PV or whatever, somatostatin, or both interneurons, or all interneurons in the brain.
Right. Yeah. So, but not to take a lot of time, but again, with duplication or deletion, it's like dozens of genes, same time changed, and when we work with one gene, yes, that's what you just mentioned. It's really sort of logical and straightforward. Anyway, it's great. This question for next 30 years of your research career at the very least. Great job. Just very quick question. Did you see any time age-dependent changes in the 16 duplication or deletion models, in terms of social interaction? How early could you, mice don't really play as rats, but still, Jackie claimed that you could study play behavior in mice. Did you try it to do that?
No, definitely never looked at play behavior, but I've done social tests on these mice at various points throughout their development, and I've been able to see pretty consistent social deficits at 6 weeks, as early as 6 weeks, I would say, and as late as like 12 weeks is probably the latest I tested.
Okay, good, excellent. Thank you.
Shane, you have a question?
Terrific job there, Ben. Really happy that I was able to take some time out of my day and watch this here. So I have a couple, well, really just one main question that I want to ask you. You mentioned you used a plus maze assay to gauge and to quantify social interaction. I know you mentioned another assay that was performed that was similar with like the cups. I don't recall the name of that. But I'm curious if there are assays that currently exist that hone in on vocalization of mice. If not, do you think that this could be a feasible option in determining social ability? Because I'm just thinking of a couple students that I have that are on the spectrum, some of which are nonverbal and, just very few vocalization. And then other students, they talk and they talk and they talk and they don't, pick up on those social cues when it's been too much. So I'm curious about vocalization, and I don't know a ton about mice. Is that their main way of communication? Is it pheromones? Is it vocalization? What is it? So interested to hear from you.
Great question. Yeah, so mice do, supposedly communicate via ultrasonic vocalizations, so that we can't hear them, but they are out there, and there are instruments that can detect them. And even there are some figures in papers where they can detect like the tone, so you can see like a specific like shape of a ultrasonic vocalization. It's like, let's say it goes like this, right? It starts at this tone and then it goes down and it stays at that, or it's up and down, whatever. And they've been able to link those specific patterns of vocalizations with different kind of functions or meanings, maybe. Actually, that may be over extrapolation, but at least they've been able to see that certain of those vocalizations are lost or reduced in specific mouse models of autism, including the 16p deletion mouse model. So yeah, that is a very feasible way of evaluating sociability. But like I said, since it requires specific equipment, that's something we don't do in our lab, but it is a neat thing.
Do you think it would be something that, down the road, you can maybe look into, or is it exclusive for that deletion mouse?
Oh, definitely not exclusive. There are multiple models that have shown that, but yeah, it definitely could be something to look into with duplication. Particularly, I'd be interested to see if, the NPAS4 rescue also affects that. That'd be pretty neat to see.
Terrific. Yeah. All right. Well, thank you so much.
Thank you, Shane.
Hey, Ben. For your RNA sequencing data, have you thought of doing ingenuity pathway analysis to figure out downstream mediators or upstream mediators of NPAS4?
Yes, I definitely thought of that. At one point, I downloaded the software, and then there was something wrong with the key code or something. I couldn't get it to work, and so I never ended up doing it. But definitely thought about it. Would be super curious to see, especially because, like I said, that HDAC5 molecule that I'm testing because it's upstream of NPAS4. I found a paper where they sequenced, basically they determined all the genes that HDAC5 regulates, and I found that 12 of those genes that HDAC5 regulates are all downregulated in my sequencing data, and none of them are upregulated, which suggests that HDAC5 might be kind of a central hub affecting not only NPAS4, but also a bunch of other genes. So I've been thinking it'd be cool to do IPA to test whether, there's some HDAC5 related pathway, or I don't know. I guess I don't really know enough about IPA if it's gene or molecule specific versus pathway specific, because, activity dependent.
With IPA, you can find out the pathways, and then you can link specifically to certain genes because it does a huge database crawl of all the genes that are important for specific pathways. So
if you have that set up, let me know. I'll be happy to send my data your way and check that out. That'd be cool.
I don't have a license, unfortunately. The licenses are expensive through QIAGEN.
Yeah, if anyone here has a license, email me, please.
Okay, so if there's no more questions from the general public, maybe we should end the session and then start the committee question.
Great job, Ben.
Thank you, Steve. Thank you for coming, everyone.